Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I want to look at histology of the pancreas, and that is part of your Module 5 hormonal communication topic. So let's just remind ourselves about homeostasis, where we need to maintain certain factors within a very narrow limit. Now, our blood glucose concentration is kept between 4 and 8 millimoles per litre of blood. Now, this is monitored very closely by the pancreas, and the pancreas is involved in the regulation of that blood sugar levels. Now, there's a very important reason for that. Glucose, we know, is necessary for glycolysis to occur. It is the respiratory substrate of choice for most organisms. So we have some tissues that are entirely dependent on the blood glucose levels so that they can then utilize that in the process of glycolysis all the way through to oxidative phosphorylation. That's because they can't store the glucose, there's no glycogen stores. They also are not able to metabolize fat. So because of that, the blood glucose concentration needs to be maintained within this very narrow limit. Also, we've got to remember that glucose is dissolved in blood plasma. It's also dissolved in our cytoplasm, dissolved in our tissue fluid. So therefore, if we have too much of it, it's going to have an effect on the water potential surrounding the tissues. Too little, again, is going to have an effect on the surrounding tissue concentration. So therefore, we need an organ that is going to monitor and regulate it, and that's the pancreas. Now, the pancreas is actually split into two regions. It's split into the exocrine region, which has the pancreatic duct, and it has the islets of Langerhans. Now, the islets of Langerhans are basically like little islands that were discovered by Dr. Langerhans, hence the name. So the islets of Langerhans then are the ones that we are really interested in when it comes to our blood glucose concentration. And the islets of Langerhans are actually made up of two types of cells. They're made up of beta cells that secrete insulin, and they're made up of alpha cells that secrete glucagon. Now, I love the alphabet in helping me with my little hints. So I always remember that glucagon has got an A in it, therefore that is the one that is secreted by my alpha cells, my A's together. So my alpha cells are going to secrete my glucagon, and I say it that way specifically as a way to help me. It's not glycogen. Do not get that confused. It's really important you learn the spelling. Glucagon. Glucose, agon. So glucagon is secreted by the alpha cells, and the beta cells therefore secrete the opposite hormone, which is insulin. Now, surrounding the cells, what we have is the fine branches of what's known as the pancreatic duct. We have acinar cells, and those acinar cells are the ones that are going to produce enzymes. Now, you don't need to know too much about this if you are doing OCRA. However, they do love to throw a little curveball at you. So the exocrine region is the cells that are going to secrete digestive enzymes, in this case from the pancreas, into a duct, and that duct will then lead down to the small intestine. If they give you this as a question, they will give you some information, and the key thing to note is that the secretion, whether we're talking about the secretion of hormones or whether we're talking about the secretion of the digestive enzymes, they may be looking at you understanding it's to do with the cytoskeleton and vesicles fusing with that membrane. So always trying to find your links. Well, the key thing that they may ask you to do in the exam is looking at the histology, and they love giving you a picture of the view down the microscope. Now, one of the key ways to note whether you're looking at the islets of Langerhans is through the differential staining. So the acinar cells, the ones that are part of the exocrine region of the pancreas, are the ones that are really dark in color. So they've got the pink and the purple color. The islets of Langerhans, they actually have a different type of staining, so they're much lighter in colour, and that therefore is a way of us identifying between the islets of Langerhans and the exocrine region. So they'll either give it to you as a picture that has lots of colour, or what they might do is they might actually give it to you in a black and white picture. If they're giving it to you with black and white, they will have the grayscale that the lighter islets of Langerhans will be obvious. So you're looking for the lighter region, and the lighter region is representing where the islets of Langerhans are found. Your alpha and your beta cells are located within the islets of Langerhans. So I hope you've liked this video, and if you have, please do click on my like button and subscribe to my channel. It helps me to be able to create more resources for you. Also, if you haven't done so already, do check out my revision platform, www.aiqchat.com.